Hey guys, welcome back to my channel Biomics. So in this today's video, we are going to discuss about another one DNA repair mechanism, that is base excision repair. So what happens, there are certain changes in the nucleotide or certain changes in the bases in the DNA, due to which there can be mutation. So due to deamination, the cytosine is converted into uracil, but there is no uracil in the DNA. So this kind of changes needs to be recognized and they needs to be repaired. This kind of mutation or this kind of changes is recognized by DNA enzyme, which is known as DNA glycolase, and it is being repaired by base excision repair. So in the second point B, we can see that this guanine is converted into oxoguanine. How? By depurination of guanine by hydrolysis creates a purine deoxyribose, which is known as OXO-G. Okay, in the same way, this 5-methylcytosine is converted into thiamine by deamination. So due to which there will be changes in the nucleotide when there will be replication, which will cause mutation in further generation. So there are certain enzymes which are working for this. So here we can see that there is involvement of two enzymes, that is the glycosylase and AP endonuclease. There are certain bonds which are present in the nucleotide. Like we can see here that there is a ribose sugar. Here we can see the purple one is a ribose sugar, and attached to it is a phosphate bond and a normal base. This base can be purine as well as pyrimidine. It can be cytosine, thiamine, uracil as well as guanine. So here with this phosphate bond, this phosphate group is attached, it is by the phosphodiester bond. It is with the phosphodiester bond. And the normal base which is present, it is by the it is attached by the glycosidic bond. So this glycosidic bond is being cleaved by the enzyme glycosylase. It recognizes this glycosylase, it recognizes the changed base pair. The base pair which is being modified, it is being recognized by this glycosylase, and the glycosidic bond is being cleaved. And due to this, there will be involvement of some other enzymes which is known as AP endonuclease. So it will cleave. After the cleavage of this bond, due to the glycosylase activity, this is being removed, this portion is being removed, this purine or apyrimidine is being removed, due to which here there is a site which is known as AP site. AP site means apurine or apyrimidine site. After this, another one enzyme AP endonuclease comes and bind. What does AP endonuclease does? It comes and bind in the AP site, apurine or apyrimidine site, and it cleaves the phosphodiester bond. Okay, due to which there will be creation of 3' OH group for DNA polymerase 1. After this removal, there will be a nucleotide which will be free. So there will be a gap which will be present in the DNA. So this gap should be filled, and it will be filled by DNA polymerase 1. Like for example, we will see this example of OXO-G. Here the guanine is present. In guanine, it base pairs with cytosine, and due to oxidation, that is some changes like OXO-G. Okay, due to which there is distortion. So the glycosylase enzyme, it scans the DNA and it recognizes this OXO-G because it is having one distortion. So what if there will be no changes of this OXO-G? That in future there will be a mutation. So this OXO-G will be replaced. It can be removed by two mechanisms. Like one is that it will be repaired by the base excision repair. Another is the failed shift glycosylase. So what happens? Guanine was base pairing with cytosine normally, but due to OXO-G, this OXO-G will base pair with adenine. It will base pair with adenine. So the thing which can be done is this adenine will be removed by failed shift glycosylase, and it will be replaced with cytosine. So here there is no changes of this nucleotide. The other thing is that it will be removed, and it will be removed by the base excision repair, and there will be correct nucleotide. Now let us see how the work of base excision repair is taking place. Here there is involvement of two enzymes like glycosylase and AP endonuclease. it recognizes this glycosylase, it recognizes the modified base, and it cleaves the glycosidic bond. After the cleavage of the glycosidic bond, there will be a purine or a pyrimidine site. Here there is no, here adenine is present, but there is no uracil. Here thymine should bind, but here uracil is binding due to certain modification. So here uracil, so here the uracil will be removed, so this place is blank now, this place is blank now. So this is being recognized by AP endonuclease enzyme. So this endonuclease, what is this? It cleaves a phosphodiester bond. It cleaves the phosphodiester bond. Phosphodiester bond is being cleaved. Okay, and there is a gap which is present at it creates a nick, and this nick is being fulfilled by DNA polymerase 1. DNA polymerase 1. Okay, as it is a short strand of DNA which needs to be synthesized, so this work can be done by DNA polymerase 1, and DNA ligase which is present, it seals the DNA. It seals the DNA. Okay, this was in case of prokaryotes. This base excision repair takes place both in case of prokaryotes as well as eukaryotes. Now let us see what happens in case of a eukaryotes. Here the glycosylase is working. Okay, here the glycosylase is identifying which base pair is being mismatched, or we can tell that which base is being modified, and here we can see that there is a short pathway, patch pathway, and long patch pathway. The short patch pathway is done by prokaryotes also. It is in both prokaryotes and eukaryotes, but there is only changes of some of the enzymes. Like in first is the incision pathway and the lyase pathway. The short patch pathway is known as the lyase pathway. So what happens here? There is involvement of DNA glycosylase till this. Here there is a purine or a pyrimidine site. Next, this nucleotide will be replaced by DNA polymerase beta. Okay, here there is involvement of a purine endonuclease 1 which is cleaving the DNA, and it has been fulfilled by DNA polymerase beta. DNA polymerase beta works for short patch pathway. Okay, and that is sealing of this DNA by the help of a enzyme or ligase enzyme which is known as Lig3 or ligase 3 in case of prokaryotes, or we can tell in case of short patch pathway, this Lig3 enzyme is used for doing the work of ligase. Now let us see the incision repair pathway. This APE1, what is this? It creates this, it cleaves the phosphodiester bond. Then we can see this light blue color. It is now like a flap. After the breakage of this phosphodiester bond, this is being removed as a flap, and the patch which is present, it is being sealed by other enzymes which is known as delta and epsilon. Delta and epsilon comes and work here. So this gap is being filled by DNA polymerase delta and epsilon in case of eukaryotes. Okay, next the work of FEN1. Here there is a involvement of other enzyme which is known as FEN1, which is known as flap endonuclease. FEN1, it is known as flap endonuclease. This flap endonuclease, it removes the flap. This flap needs to be removed because there is synthesis of this DNA due to polymerase delta and epsilon. So after filling this will be resealed, and this flap will be removed out by the involvement of Fe1 endonuclease, and here there is involvement of Lig1 for the ligation in case of eukaryotes. So what happens in case of prokaryotes? The AP endonuclease as well as the DNA polymerase 1 was working. In case of eukaryotes, DNA polymerase B is working, and for resealing Lig3 is working, and in case of eukaryotes, for filling the gap, delta and DNA polymerase delta and epsilon are working. For removing the flap, FEN endonuclease is working, and for sealing the gap, Lig1 is working in case of prokaryotes, or in a long pathway, or we can tell that incision pathway. This was all about the base excision repair pathway in case of prokaryotes and the eukaryotes. Thank you for watching this video till the end, and if you like this video, please do like, share and subscribe. Thank you.